In this segment, we will discuss the methods which are used to purify the antibodies. You know, the antibodies, they are extremely important in determining the biological system, the biological activation, and also the biological methods of estimation. Now we will study that how antibodies can be purified and stored in this segment. Now antibodies, they can be purified from serum by the addition of chiotropic ions in the form of saturated ammonium sulfate. Now what are chiotropic ions? Chiotropic ions are those ions which disrupt the hydrogen bonding of the water molecule. You see, when they are added to the water, they are the ions that the hydrogen bonds which are present in the water, that is dissociated, that is separated, and they are known as the chiotropic ions. Ammonium sulfate is one of the chiotropic chemical which are used for this purpose. You know that alcohol is also chiotropic, but it works in a different manner. So what we do, that ammonium sulfate preferentially precipitates the antibody fraction in around 60% saturation. That is, when we use ammonium sulfate for the purification of antibody, it can purify it up to the 60%. It provides rapid method of IgG, which stands for immunoglobulin G antibody purification. And that is the one method for the ammonium sulfate. You will recall that, as a matter of fact, ammonium sulfate is also used to precipitate the proteins which are present in the solution. Now this method, mind you, does not work well in the tissue culture supernatant. That is, if the antibodies or if these things are in the tissue culture supernatant, ammonium sulfate shall not work for the purification or for the precipitation. Now how ammonium sulfate precipitation works? Now ammonium sulfate precipitation can be used as a preparatory method. You see, it is a method which separates, which precipitates the crude antibodies, and this is used prior to the further chromatographic purification. That is, when we have to purify the antibodies, initially we use ammonium sulfate, which precipitates the antibodies, but in the crude method, and further purification is done by chromatographic techniques, by chromatographic purification, which yield us the purified antibodies. Now, there are very many devices which are used, as a matter of fact, to purify the antibodies. Now, one is the tangential devices and the centrifugal concentration. Now, these devices, tangential flow devices, are that the serum or the medium is allowed to pass in a horizontal way, in a tangential way, or the tangent to the membranes where it is purified, or by the centrifugation method where these proteins or these antibodies are centrifuged. And that when these methods are used, what happens, that it may be used to reduce the volume of 10% of the starting amount. You see, the starting amount, it reduces 10% less, and then ammonium sulfate can be used. Now, this makes antibody purification by affinity chromatography much easier with the smaller volume of the liquid. That is, if we have to use the tangential flow method or the centrifugation method when we use the affinity chromatography. So you see, in this diagram we have shown that one is the column and the solution is added. Then in the second, the antibodies is actually absorbed on that, and eventually in the column we elute it with a buffer or with the special elutes which remove the antibodies from the column, and it is collected there. Now, antibodies from both polyclonal and the monoclonal sources can be purified by similar means, that we use the columns for the affinity chromatography, and then that the antibody which is absorbed on the column is eluted and further purified. Now, immunoglobulin G, this antibody type is immunoglobulin G, which allows the purification by the protein A and G affinity chromatography. That is, the IgG, immunoglobulin G, can be purified by the protein A or G affinity chromatography. Now, protein A and G, let's see, they are the derivatives of bacterial cell and have the ability to reversibly bind IgG molecules. That is, the immunoglobulin G molecule, they can be bound with the protein A and G, but at the same time with a specific elute. Elute are the solution which are used to remove the antibody attached to the column or to the affinity chromatography to remove. So they can, it is reversibly bound with them. Binding to the column occurs at the neutral pH, and the pure antibody fraction can be eluted at pH 2, that is the method. Now, fractions are collected and neutralized back to pH 7, that is 2 and the 7, and antibody containing fractions are identified by spectrophotometry using the absorbance at 280 nanometer. Now, solution of protein at 1 milligram per centimeter cube will give an absorbance reading of 1.4 at 280 nanometer, and purified antibodies should be adjusted again to the 1 milligram per cubic centimeter and kept at 4 degrees centigrade or minus 20 degrees centigrade for long-term storage. So, the purified method, if we have to use, we can store them at 4 degrees centigrade if we have to use them immediately, or can store at minus 20 degrees centigrade for long-term storage, so that they can be utilized for further experimentation in our molecular biology experiments.